ABSEC's online support program, Class 9th, Week 9, Lesson 3, Subject Biology. Textbook Biology, page number 87 to 88, Unit 4, Cell Cycle, topic Cell Cycle. Objectives. By the end of the lesson, students will be able to revise cell cycle, two phases of cell cycle, that is mitotic phase and interphase, interphase subdivision, that is G1 phase, S phase, G2 phase, G0 phase. Have a look on the flow chart of the cell cycle. As I have told you that cell cycle is divided into two major phases, that is interphase and mitotic phase. Interphase is further subdivided into G1 phase, S phase, G2 and G0 phase. Mitosis phase is subdivided into two phases, that is karyokinesis and cytokinesis. Karyokinesis has its own steps, that is prophase, metaphase, anaphase and telophase. But today we will be discussing the interphase. Cell cycle. The definition of the cell cycle is that cell cycle is series of events from the time a cell is produced until it completes mitosis and produces new cell. It means that when a cell is dividing into two daughter cells, so in between there are a lot of steps which has to be performed by a cell. Now the events that are taking place in between from the cell division up to the cell formation, these events are actually named as cell cycle. And then how the cell completes its division? It completes its division with the help of mitosis and the cell divides into two daughter cells. We are having two phases of cell cycle, that is interphase and mitotic phase. Mitotic phase. If we talk about the mitotic phase, so it is a short period of cell cycle, and interphase, it is a longer period of cell cycle. A lot of time is required for interphase. Why? Because here a cell prepares itself for cell division, and the metabolic activity of interphase will be high as it performs its various functions. It means that here proteins formation take place, enzymes are synthesized, organelles are produced, the cell grow in size. For this purpose, its metabolic activity will be very much high. Just have a look on this diagram or a pie chart. You can see this G1 phase. G1 is also called a gap phase. When a cell enters into a gap phase, from gap phase it moves towards the S phase. S phase is also called as synthesis phase. Why synthesis phase? Because here DNA is synthesized. Then after this it will move towards the G2 phase, which is also called as gap 2 phase. After this it will enter into M phase, which is called mitosis, and here actually the cell division is taken place. First we will be discussing the G1 phase. Here are four important points of the G1 phase. First is that cell increases its supply of proteins. As cell is preparing itself for cell division, for this purpose it will be needing proteins. So the supply of the proteins towards the cell is increased. Second is that it increases the number of organelles, that is mitochondria and ribosome. Beside these two, we are also having endoplasmic reticulum, ribosomes, lysosomes, centrioles. They are performing different functions inside the cell. if I talk about the mitosis, so mitosis is called the powerhouse, right? It helps in the production of ATP, and ribosomes help in the protein synthesis. So each and every organelle is performing its specific function. So when the cell is preparing itself for cell division, so what will happen, that first the supply of the protein will be increased. Secondly, it will also increase the number of the organelles. As the supply of the proteins and the number of organelles are increased, so automatically the cell will grow in size. And at the last, various enzymes are synthesized that are required in the next phase. It's mean here enzymes synthesis will take place, but those enzymes will not be required in this phase. It will be not used in this phase, but they are only synthesized in the G1 phase, and then they are used in the next phase, that is the S phase. Then S phase. What happened in the S phase, as S phase is the second phase of the interphase. Here simply the cell will duplicate its chromosome. As you can see in the diagram that a single chromatid is present. But when this chromatid is duplicated, so it will form a full chromosome. And a chromosome consists of a two sister chromatid. And you all know the function of the chromosome, that it helps in the transfer of the genetic material. The two sister chromatids are held at a point that is called a centromere. So you can see that cell duplicates its chromosomes. As a result, each chromosome consists of two sister chromatids. Then we are having the G2 phase. What happened in the G2 phase? Simply the cell will prepare all that proteins which are essential for mitosis. So which proteins are essential for mitosis? That are the kinetochore proteins. Why kinetochore proteins? Because it helps in the production of the spindle fibers. What is the function of the spindle fiber? Spindle fiber help in the attachment of the chromosomes. It helps in the pulling of the chromosomes, and it also helps in the separation of the chromosomes. So here in G2 phase, all those proteins will be produced, or they will be prepared, which are essential for mitosis. And I have given you the example of the kinetochore proteins, which help in the production of the spindle fiber. After all of these phases, what will happen? Now the cell is fully prepared to enter into the M phase. What is M phase? M phase is the mitotic phase. It is that phase where the cell division takes place. And here a single cell will be divided into two daughter cells. Now at the end, I will be telling you about the G0 phase. What is the G0 phase? G0 phase is that phase where a cell will permanently or temporarily enter, but only those cells which are not able to divide or which stop dividing itself, so they will simply enter into the G0 phase. We are having some kind of a cells that enter into the G0 phase either permanently or temporarily or for the indefinite period of a time. If I am talking about the multicellular eukaryotes, so from the G1 phase they will directly enter into the G0 phase. Only those cells which are not dividing or which stop its division. So here are some examples. Some cells enter G0 phase for indefinite period of time, that are the, or for example, the neuron cells. Neuron cells are actually those cells which are present inside your brain, right? And they will enter into the G0 phase for indefinite period of time because there is no need of division of those cells, so they enter into the G0 phase. For indefinite period of times means like we don't know that by which time it will enter into the G1 phase for further division. Then are some cells enter into the G0 phase semi-permanently, example some cells of the liver and the kidney. Liver and kidney cells enter semi-permanently into the G0 phase because a time reached that not need that it do not need any kind of division. The cells are not divided, so they enter into the G naught phase. Then at the end, I'll be telling you about those cells which never stop dividing or which never enter into the G naught phase, and those are the epithelial cells. As I told you in the first lecture about the epithelial cells, that epithelial cells are actually protective in function. They produced a lining in the esophagus, in the cavities, in the alimentary canal, and they serve the function of the protection. So these cells never enter into the G naught phase, and they will divide throughout the whole life. So this was all about your today's lecture. Hope you find it easy and interesting. Thank you.